Okay, today we'll have an introduction to bacterial genetics, very general, but giving an overview of this whole huge field. Now, bacteria are very cool and interesting in and of themselves, but of course they have a lot of importance to us because they include a lot of human pathogens. We're also going to talk about the what I call genetic entities, plasmids, phages, and transposons that inhabit bacterial cells, partly because they're interesting also, but also because the genetic differences that distinguish pathogens from non-pathogens are often the result of genes that were carried by these genetic entities that distinguish those types of cells. Okay, so in general, bacteria do not have meiosis, but they do have ways of exchanging genetic information from one cell to another, sometimes from one species to another. Okay, this will often involve plasmids and viruses, but chromosomal DNA can also be transferred. Okay, so the general term horizontal transfer refers to the fact that DNA can be exchanged not just from one cell to another, but from one organism to another. Okay, generally speaking, they have a single circular chromosome, which consists of double-stranded DNA, and the genes number in the thousands. Now, the organization of their genome in one single circle versus our multiple chromosomes does distinguish them from eukaryotes, among many other differences. Okay, certainly they grow by binary fission, and in terms of the lab, we will grow them either in liquid broth cultures or on agar-containing medium in Petri dishes. So we will be looking at colonies to do some of our genetic analyses. Okay, here's a picture of E. coli cells. They're very small and very oblong, and they kind of look like little globs. It's kind of hard to really imagine how complex these very small and simple organisms really are genetically, but when you think of what they have to do metabolically, you can account for several thousand genes that make up their genomes, generally speaking. Here's one way of growing E. coli on a Petri dish. It's got agar in it to keep it solid, but it also has all the nutrients that the E. coli cell needs to proliferate and grow from a single cell laid on the Petri dish into an actual colony in a simple overnight incubation. Okay, we sometimes grow our cells in broth. On the right-hand side of this picture, we see what the broth looks like before we put the cells into it. And on the left, we have a culture flask where you can see that the cells in a simple overnight growth have made this solution very cloudy, and the cloudiness is due to all those cells. Okay, in terms of its genome, E. coli is a little on the big side prokaryote-wise, but not too much, about 4.7 million base pairs. Now, one important difference that we're going to have to be considering is that the genes are named according to their function. So we're not used to this kind of nomenclature, but they're all named according to what their job is to do. For example, PRD is for synthesis of pyrimidines, Arg-G for synthesis of arginine, and the letter designates the locus because each of those processes, pyrimidine biosynthesis and arginine biosynthesis, requires, of course, numerous enzymes, which are encoded by numerous genes. So the letter designates the actual gene. The other thing that seems a little odd is that the units on the genetic map are in minutes. This is because when the chromosome can be transferred from one cell to cell, it takes 100 minutes to do that. So the entire circle has 100 minutes on it, going from zero clockwise to where all the individual genes are located. Okay, so here's a representation of a harmless E. coli K-12, which lives in our intestines and causes us no problems. So notice that the map is a circle. It goes from 0 to 100 in minutes. And that some of the genes are indicated by way of a sort of genetic map, similar to what we've seen in eukaryotes. But for example, you can see there's a gene designation, LAC, for lactose at around 8 minutes. And that encodes the genes needed to get lactose, the disaccharide, into metabolism. Then we have, here's PRD, the pyrimidine biosynthesis genes. We have around the circle the Arg genes and others. So these are just markers of the several thousand genes on the chromosome, but we can use them as sort of markings of where we are in the genetic map. Okay, so the coding capacity is actually huge. Remember that these cells are amazing. They can actually synthesize their own amino acids, synthesize their own purines and pyrimidines from simple compounds placed in the medium. Of course, they need to have energy-generating mechanisms, so they metabolize sugars through the TCA cycle. They synthesize lipids. They have to synthesize RNA and protein, do DNA replication and repair, etc. So when you consider everything they have to do, a few thousand genes almost seems too few rather than too many. Okay, so in terms of the genetic entities that do the real work in bacteria, these are plasmids, viruses, transposons, and IS elements. Now these inhabit the bacteria, and they rely on the cell for their propagation, but they do usually have their own genes, a small number of them in each case. And such things are responsible for transfer of bacterial genes from cell to cell, which is what in bacteria comes closest to sexual reproduction. Okay, so just to contrast a pathogen from a non-pathogen, we have E. coli 0157H7. Okay, this is a very severe infectious agent. It causes hemorrhagic colitis, which can be deadly. But these are not innately E. coli genes that cause this. This particular strain of E. coli has a lot of associated virulence factors and toxins that they produce, which were carried by phages to the non-toxigenic strain, but now are part of this particular strain's genome. So the changes involve the phages, plasmids, and transposons that actually carry these kinds of things. So here's a genome comparison of this toxigenic strain to our normal E. coli K-12. In the circle, what's shown in blue is the genes, the regular E. coli MAC genes, that are present in both of those strains. Now, the areas that are shown in red are characteristic of the harmless E. coli K-12, but the areas in orange are specific to the pathogen, the 0157H7. So this species definitions are a little bit difficult in bacteria because you do have these massive differences between strains of essentially the same species. So in terms of genetic analysis, we're going to have to think about what you call a phenotype. Now, they don't have curly wings, they don't have different color eyes, they don't have other things, but we do have phenotypes that we consider as phenotypes that are characteristic of these organisms. And the ones that we're going to be using for our purposes are prototrophy versus auxotrophy, carbon source utilization, resistance and sensitivity to antibiotics, and sometimes resistance to things like cold, heat, and other chemicals such as salts. So they're not phenotypes in the visible sense, but they are things that we can observe by growing the cells in particular types of media or under particular conditions. Here's our E. coli K-12 again. Right, so a wild type E. coli is one that can grow on what we call a minimum medium. Now, the minimum medium contains a carbon source, glucose for example, a nitrogen source, ammonia, some vitamins, some minerals, etc. But from these simple compounds, the cell can synthesize all of its components and obtain energy for their own reproduction. So there's many important biochemical pathways which are essential for the growth of the E. coli cell. And the wild type E. coli is capable of all those things. Now, auxotrophic versus prototrophic. What we're going to do is compare a particular mutant strain, an auxotroph, versus the wild type, which is a prototroph. Okay, so prototroph means that the cells don't need any extra nutrients beyond what the wild type cells need. They have all the pathways that they need to make new cells. Now, auxotroph means that the cell needs something extra because they have mutations in one or more essential pathways. As a result, they cannot grow on minimum media. But they can grow if the media also contains the component that they can't synthesize for themselves. And that's going to become important in a few minutes. Now, what we're calling rich medium includes all the amino acids, purines, pyrimidines, plus, of course, the nitrogen and carbon. And both prototrophs and auxotrophs can grow in this, because the auxotrophs are missing, for example, some amino acid biosynthetic pathway. But if we put in the amino acid to the medium, the auxotroph can grow and be supplemented by that. terms of gene names, the genes are named according to what they do, and this is a simple example of proline biosynthesis beginning with glutamate. So we have glutamate at the beginning, and a series of genes encode a series of enzymes which sequentially convert the glutamine to proline. So that's Pro-B, Pro-A, and Pro-C. So those three enzymes are encoded in the genome that enable the cell to produce its own proline from glutamate. Now, a cell that's missing any one of these enzymes would not be able to synthesize proline, and so that would be an example of a proline auxotroph. But that cell, therefore, could not grow on minimal medium, even though it's got glucose or ammonium, because it needs proline for the synthesis of its proteins. But we can supplement the medium with proline and enable the cell to grow even though it's actually missing a complete biochemical pathway for that amino acid. Similarly here, we have the pathway that goes from glutamate to the synthesis of arginine, and there's a whole slew of enzymes, all called Arg something, A through H, that are required for that process. So again, a mutation in any one of those genes that leads to a defective enzyme would cause the cell to become an arginine auxotroph, in that it would not be able to create its own arginine and will require that supplement in the medium in order to be able to grow. Here's another picture of arginine biosynthesis. Again, this is the last few steps, encoded by the genes Arg-E, Arg-F, Arg-G, and Arg-H, which sequentially convert N-acetyl ornithine to arginine. So a mutation in any one of those would cause the production of an arginine auxotroph, which could grow if arginine is present in the medium, but cannot grow if it's absent. So I think that's what I just said. If we add arginine to the minimal medium, the auxotroph can grow, so we can provide them with the essential amino acid. They will take it up from the medium and use it. Okay, here's a clicker. An Arg-mutant can grow on minimal lacking arginine, synthesizes arginine from simple precursors, is killed by arginine in the medium, lacks an enzyme for arginine biosynthesis, and does not need arginine for the synthesis of its proteins. Okay, so the answer is D. An Arg-mutant lacks an enzyme for arginine biosynthesis. Here's some examples of what this looks like if we're going to be comparing prototrophs and auxotrophs on rich and minimal medium. Okay, so this is supposed to be a rich medium, and the little dots are colonies where we placed different strains of E. coli as little dots on the medium and let them grow overnight. On the rich medium, everybody grows. On the minimal medium, which does include glucose, but no extra amino acids, we can see that some of the strains are able to grow just fine, but the three empty circles imply that there were cells placed there, but they were not able to grow. So those three cells or colonies are auxotrophic, and they require something that the wild-type strain does not require. So we can figure out some of what that is by placing the same cells on a minimal medium plus leucine or plus tryptophan. So if we take these auxotrophs, these same ones, well, we've got three auxotrophs here, the little empty colonies, and place them on medium with leucine, we can see that the one on the upper right is now able to grow, whereas the other two are not. Similarly, we've got the minimal plus tryptophan. The one on the lower left is now able to grow, whereas the other ones are not. So this tells us some important things about the genetics of those particular strains. So hold on to this idea. The minimal plus leucine means that that cell that was an auxotroph requires leucine because when we supplement it, it's now able to grow. So this strain itself is Lu minus, and it grows on media with leucine. So this is a Lu minus auxotroph. Same for the tryptophan, that cell can now grow if we give it tryptophan. So that is a trip minus cell, trip minus colony, which can grow if we supplement tryptophan in the medium. Now the other thing to think about here is that if we're considering leucine and tryptophan as the possible auxotrophies of these particular strains, that this Lu minus cell, which requires leucine to grow, is actually also trip plus. And the trip minus mutant is also Lu plus. And the reason that's true is that this auxotroph grew if we added leucine and only leucine to the medium. So that means that's the only thing it requires. Only leucine is its only auxotrophy. Same with the trip. It's trip minus because it requires tryptophan to grow, but it doesn't require leucine because the only thing we put in here was tryptophan. So it's genotypically going to be trip minus Lu plus in that it can be supplemented with tryptophan in and of itself. So this is pretty much what I just said. So the different plates test for the compounds that each of these particular cells might need. So we can see that the cells do not grow on the minimal, so they need something, but we don't know what that is. And then the combination of plates is going to tell us what exactly it is that they are missing. Right, so the plus and minus can be a little confusing here. A Lu minus mutant rather, is one that requires leucine. So the medium is plus leucine, right? So the one that's Lu minus requires leucine and only leucine. So we're going to have some examples where we have to actually figure out some slightly more subtle ways of figuring these things out. Okay, so the other mutant, which was trip minus, we concluded that that's Lu plus because it required only tryptophan, did not require leucine. Now notice that the other ones didn't grow on either of these, so we don't know what they require. We know they require something, but either Lu or trip by itself is not sufficient to grow those. So those remain unknown auxotrophies from the plates that we looked at. Okay, our mutant strain is Arg minus, Pro minus, therefore, the cells need arginine and proline in the medium to grow. The cells do not need these amino acids to grow. The cells cannot grow on minimal medium. A and B are true. A and C are true. Yes, A and C are true. They need arginine and proline, and they cannot grow on minimal medium. Okay, the other phenotypes that are of interest and important are carbon source utilization is one of them. Okay, so this is confusing in that it's different, sort of the opposite from the amino acid auxotrophies. The usual carbon is going to be considered glucose, but other carbon sources such as lactose, raffinose, and galactose can be used. A wild-type E. coli cell is Lac+, meaning that it can grow if given only lactose. A Lac- mutant cannot grow with this sugar. It can't use it. It's not like it requires lactose. It's the opposite. It cannot use it because it does not have the enzymes it needs to break down the lactose disaccharide into the two monosaccharides that can then go into glycolysis. So it's sort of the opposite. A Lac- mutant cannot grow on lactose as a sole carbon source. So here we go. Auxotrophs require something. They can grow on minimal if it's provided whatever their auxotrophy is. The carbon source can't grow on something. Lac- mutants require other sugars, glucose. They will not grow if the medium contains only lactose. So they do not grow on min plus lactose, or rich medium where lactose is the only carbon source. A mutant is Lac-, therefore A requires lactose to grow. It does not use lactose and cannot grow if lactose is the only carbon source. So the answer, of course, is B. Keep that in mind. Our strain is Arg-, Lac-. Therefore, we need arginine and lactose in the medium for it to grow. We need arginine and glucose in the medium for it to grow. We need to have a medium that lacks both of these for it to grow. We need to have lactose only for it to grow. Okay, so the answer is B. We need arginine and glucose in the medium for it to grow, not lactose. Okay, another important phenotype is drug sensitivity versus resistance. So wild-type E. coli is sensitive to many different drugs. That means that they die in their presence. But resistance very often occurs in the bacterial world because of genes that are transferred by plasmids and transposons, and sometimes phages that inhabit bacterial cells. So whereas the wild-type E. coli might be sensitive, it can carry drug resistance, largely as a result of having transposons and things in their genome. So the medium for selection here, the easiest, is rich medium plus the drug. So that way you're going to supplement any auxotrophies you have. Resistant cells live to form colonies. Sensitive ones die. So usually just sensitive versus resistance you determine by plating the media, the cells on the media with the drug. So growth means that they're resistant. No growth means that they're sensitive. Now it's important to recognize that any auxotrophies could confuse the issue if you were to use minimal medium for this experiment. So to look for drug resistance, it's best to use a rich medium where any auxotrophy has been supplemented already. Okay, for some reason the resistance genes don't have letters, names. They have a superscript, ampicillin superscript R versus ampicillin superscript S. There are other ways of describing the ampicillin resistance gene, not just AMP-R, but this is the way I like it. Okay, now we're going to look at the worksheet. Okay, here's our first worksheet having to do with auxotrophies and other things. And what you have to do is fill in the table in the top by comparing the growth of these different mutant strains on the different types of media. Okay, so we have each plate's divided into quadrants. We have wild type in one, and then mutants 1, 2, and 3 around the circle. And each one is going to be grown on a variety of media, and from this information we're supposed to infer the genotypes of those organisms. Okay, so the little black triangle implies that growth occurred, and the blank spot means that it did not. Okay, so the first thing we need to do is let's look at the streptomycin, because that's the easiest. The rich medium plus glucose and streptomycin. The wild type did not grow, which is normal. The mutant one did grow. So mutant 1 is streptomycin resistant, and the other two are streptomycin sensitive. Now notice we can tell this because the rich medium will supplement any auxotrophies, so the only thing in play here is really whether the mutants are sensitive or resistant to that drug. Okay, the next plate is rich medium with lactose as a sole carbon source, and now we're going to look to see if the cells are lac plus or lac minus. So remember that lac plus means the cell can use lactose as a carbon source, not that it requires it. Okay, so the wild type is lac plus. The first mutant does not grow, so that one's lac minus. Mutants 2 and 3 do grow, so those are lac plus. Okay, so we'll fill those into the table on the next slide. Okay, the next little petri dish is minimal medium only, and we see that the wild type grows, of course, as does mutant number 1, but 2 and 3 do not grow. Okay, so what that means is that the mutant 1 does not have any auxotrophies at all. It's able to grow on the minimal medium, assuming glucose is present, perfectly well. So mutant 1 we can fill in the table is Lu plus, His plus, Ada plus. No problem there. Okay, the next one, minimal plus leucine, histidine, and adenine. Okay, so from the previous minimal plate, we learned that mutants 2 and 3 have auxotrophies of some kind, which is to say that they require something that the wild type does not require. And we learn what possibilities there are from this plate because minimal plus leucine, histidine, and adenine, both of those mutants do grow. So we know they have auxotrophies, and we know that they have one or more of the leucine, histidine, or adenine. It could be only one, or it could be some combination of those. All we know right now is that combination allows growth. So that restricts our possibilities. Okay, so the next plate is minimal plus leucine. Nobody grows. Mutants 2 and 3, neither one of them grows. Minimal plus histidine, neither one of them grows. Minimal plus adenine, mutant 3 does grow. Okay, so that tells us that the only requirement that mutant 3 has is adenine. So mutant 3 is Ada minus, but it's also His plus and Lu plus because it does not require those supplements. The only thing it needs is adenine. Okay, mutant 2 is a little bit more complicated because minimal plus leucine doesn't allow it to grow. Histidine alone doesn't allow it to grow. Adenine alone does not allow it to grow. But if you add leucine and histidine, it does grow. So that tells us that even though no single supplement was allowed the mutant to grow. None of the three was sufficient to supplement the auxotrophy. The combination of leucine and histidine does. So that tells us that mutant 2 is Lu minus, His minus, but add a plus because it does not require adenine, whereas it does require those other two supplements. And that's consistent with the rest of the plates. The mutant minimal plus leucine and adenine. Mutant 3 grows. Mutant 2 doesn't because it doesn't have the histidine. Mutant minimal plus histidine and adenine. Mutant 2 still doesn't grow. It's got histidine but no leucine. And the adenine is not going to be sufficient to allow it to grow. So it's filled in on this slide where all the auxotrophies are covered and we have the genotypes of all. So if you look at it logically, it's really not difficult. One thing to do when you're looking for auxotrophies, though, is to look for whether any single amino acid or base allows it to grow, to tell you whether it's a single auxotrophy or multiple ones. That's the only key to this, really. Okay, so we're going to have a quick introduction to the genetic entities that do the real work in bacteria, plasmids, viruses, and transposons. Okay, so the idea is that these elements contain some genes which encode functions that are important for their own transmission in life, but they rely heavily on the cell for basically everything, including protein synthesis, RNA synthesis, etc. Okay, plasmids are the most familiar. They're circular, but they vary a lot in size naturally from as little as 3,000 base pairs or less to more than 100,000 base pairs. Generally speaking, plasmids must have a replication origin to allow them to replicate inside the host cell, separate from the bacterial chromosome. So that's an important and general feature of plasmids in general. They must be able to replicate independently of the chromosome. They're very variable in what they encode, for sure, but they can confer extra phenotypes on the bacterial cell. For example, they can confer resistance to drugs because they carry transposons. They can encode actual biochemical pathways. They can do many different things. But they are heavily reliant on the host cell, for example, for all aspects, virtually all aspects of translation, and most aspects of transcription and replication. So I just said this already. They can confer new phenotypes on host cells, resistance, biochemical pathways, etc. Here's a little diagram of the plasmids in the bacterial cell, and you can see that there's different plasmids in this cell, and they are replicating as circles separately from the host cell chromosome. There are plasmids, however, which can actually integrate or join into the host cell chromosome and live as residents within the host cell chromosome. This is not an unusual feature of some plasmids. Here's a good example, the F plasmid. It's a big plasmid. It's got almost 100,000 base pairs in it, and it's able to encode its own transmission from cell to cell. So it includes genes called the Tra region, which encode the proteins that are necessary for that process, which we'll explain better next time, as well as being the host carrier for a transposon and a couple of insertion elements. So this is a big plasmid, and what it's able to do is transmit itself from one cell to the other. Okay. It does normally replicate independently of the cell chromosome, but it can also integrate into the chromosome, becoming part of the chromosome. It doesn't naturally include drug resistance, but it can carry drug resistance in the form of transposons. Here's another type of plasma which is also transmissible. It has the ability to transmit itself from cell to cell. But this is called an R factor because it contains multidrug resistance. So this is a very scary plasmid because it can transmit itself from cell to cell. But when it does that, it carries resistance to a whole slew of antibiotics along with it. So any cell that acquires the R plasmid from another cell simultaneously acquires resistance to tetracycline, kanamycin, ampicillin, hygromycin, etc. So that's very frightening in a hospital setting where you get the simultaneous drug resistance when this plasmid spreads itself throughout the bacterial population. Yeah, the most familiar plasmids are the ones used for cloning. These can replicate independently of the host cell chromosome. They do carry antibiotic resistance for experimental reasons. These are not natural plasmids, however. They've been put together from pieces and parts of other plasmids to suit the experimental needs of people that want to clone DNA. So here is one commonly used one. It's circular. It's small. It's less than 3,000 base pairs. But notice it does carry a drug resistance. It's called BLA, standing for beta-lactamase, also designated as AP superscript R. But this is the same old good old ampicillin resistance gene that we've talked about before. Also contains an origin and replication, and also contains some synthetic bits of DNA that are useful for cloning purposes. Okay, so the ampicillin resistance gene, penicillin resistance, the gene encodes a protein, which is a beta-lactamase, which is to say it can take this beta-lactam ring of the penicillin and open it up, rendering the penicillin inactive and unable to kill the cell. So the basis of penicillin resistance is basically the ability to open that up and essentially degrade the drug so that it's no longer going to cause the cell wall to get fragile. Almost always carry drug resistance, same thing. Production of plasmid-encoded proteins requires plasmid, promoters, origins of replication, ribosomes, tRNAs. Promoters. It has its own origin of replication, but that has nothing to do with protein synthesis. Ribosomes and tRNAs are provided by the host cell. Promoter, however, the ampicillin resistance gene needs to be expressed, need to have RNA production and protein production in order for this plasmid to confer resistance. So the plasmid must contain a promoter, which is recognizable by the host cell polymerase, so that the beta-lactamase can be produced. All right, next in our quick tour, we have IS elements and transposons. So these are small molecules that can move around from one location in the genome to another. This is the process of transposition. Okay, now they're unusual in that, well, different from phages and plasmids, in that they do not generally carry their own replication origin, but they can kind of hitch a ride on anything else that happens to be there. So they can be sitting in a plasmid, they can go into the chromosome if they so desire. Okay, so transposons are more complicated elements. IS elements encode the transposase for movement, and transposons include IS elements, but are also carry antibiotic resistance as a result. Okay, so they're not very big, a kilobase or so general range, and they have non-imaginative names like IS1, IS2, IS3, etc. Transposons are called Tn3, Tn5, Tn something. Okay, important to note, they're not circular, and they need to hitch a ride because they have no origin of their own. Here's a transposon Tn10 as an example. It consists of two IS elements, one on each end, and one of those encodes the transposase. In the middle, we have the tetracycline resistance gene. So this transposon will confer resistance to the tetracycline, which is a drug, when it's present in a cell, and it can move around from one location to another by means of the transposase that's carried by one of the IS elements, that's encoded by one of the IS elements. The IS elements and transposons generally contain repeat sequences on each end, and they could be pointing in the same direction or opposite. But the transposase itself is encoded by one of the IS elements. Right, so here's some examples. Here's an IS element by itself. It has these repeated sequences, one at each end, with the transposase kind of stuck in the middle. An example of a composite transposon is Tn5. It contains two IS elements, one at each end, and it's got a kanamycin resistance gene in the middle. So transposons are made of a resistance gene plus transposase encoding IS elements at their ends. Okay, so in general, they can hitch a ride at anything, and they can certainly confer advantages to the E. coli cell because they encode resistance to antibiotics. And these are responsible for the formation of the R factors mentioned before, which are conjugatable plasmids that carry antibiotic resistance genes on the same plasmid. IS elements are not rare. Here's a picture of IS elements on a typical E. coli chromosome, so there are many of them. But they don't move very often, fortunately. Okay, a few words about viruses, otherwise known as bacteriophages. Now, there's a little nomenclature issue here. Bacteriophage refers to a bacterial virus, the virus that infects bacteria. Now, there are plenty of viruses that are eukaryotic viruses, which are not bacteriophages. The word bacteriophage is restricted to bacteria. They're generally 50 kb or less, but they do encode proteins for their own survival, right? So they need to make a coat and tail capsule, regulatory proteins and things, but they will rely necessarily on the host cell for protein synthesis for sure, and most aspects of transcription, and most aspects of replication. They do replicate independently of the host chromosome during the lytic part of the cycle, but some can integrate into the host chromosome, as we will see. So lambda and T4 are the most famous. P1 is another. Right, here's a typical phage particle. Another word, virion, refers to the actual phage particle. Virion is used for eukaryotic viruses as well. So with lambda, it's a complicated phage particle. It's made of protein. The DNA is inside, and it consists of a head, collar, tail, all of which are part of this gene delivery system, really, whose job it is to deliver the phage DNA to the recipient cell so that the phage can then replicate inside that cell. Here's what bacteriophage lambda looks like. As we'll see later, sometimes it's a linear molecule, sometimes it's a circle, but this is the circular form that is shown typically. And it has sort of strange gene names, X's, and some are just given letters. It's about 50,000 base pairs totally. So it has different forms, etc.